Hello again, everyone. Today we're going to talk about Chapter 9, DNA Structure and Replication. And we're going to be looking at two sections. Section 2, which looks at DNA structure, and Section 3, which looks at DNA replication, maintaining the genetic information. Now, we're not going to go into super detail on especially the structure, and that's just because you have learned this back in biology. You should have some memory of it, some foundational knowledge. We are going to still review it, but we're not going to go into as much detail as we would in biology class because you've already passed that. All right, first let's start with structure. First thing we should talk about is, what is a gene? Well, simply put, a gene is a segment of DNA that contains the information to specify a sequence of amino acids in a protein. So what we're saying is that in your DNA, there are sections of bases, and these can be thousands of bases long or even longer than that. But these sections code for particular traits, and that's what a gene is. It's a section of the DNA that codes for a trait. Now, how it codes for that is because within that sequence of bases, it contains the information for the order of the amino acid chain that would be used to create a protein. All right, let's talk a little bit about the double helix. So DNA, as we saw in lesson 1, is known to be a double helix, meaning that there are two strands that are twisted together. So DNA double helix is a ladder of alternating deoxyribose and phosphate groups. So on the outside portions of the ladder, we have the sugar, which is deoxyribose, and phosphate. So there's sugar, phosphate, sugar, phosphate, sugar, phosphate. And on the other side, same thing. Sugar, phosphate, sugar, phosphate, sugar, phosphate. And they're connected in the middle. So if you imagine a ladder, just said the sugar and phosphate are the two sides of it. The steps or the rungs of the ladder are formed by the complementary nitrogenous bases. Now these base pairs, we said they're complementary, which means that we can use one to figure out the other based on Chargaff's rule. Chargaff's rule says that adenine will always bond with thymine, and guanine will always bond with cytosine. And that's because of their overall shape. So adenine and guanine are both purines. They are a larger double ring structure. Thymine and cytosine are pyrimidines. They are smaller structure. It's a single ring. So you always have one purine for one pyrimidine. We'll look more at this in a moment in the picture. But adenine is a purine, and it always bonds with thymine, which is a pyrimidine. Now the reason why those two form a bond and why they're complementary is the number of hydrogen bonds. They can only form 2 hydrogen bonds between each other. Whereas guanine, a purine, and cytosine, a pyrimidine, can form 3 hydrogen bonds. All right, so that's what this picture up here is showing. So these right here, this is a sugar, this is a sugar. Same over here. So sugar, phosphate, sugar, sugar, phosphate, sugar. And we have cytosine and thymine. Those are our 2 pyrimidines. Then we have guanine and adenine, our 2 purines. You can see how they are larger. So according to Chargaff, the number of purines and the number of pyrimidines equal each other, which means that you always have a purine and a pyrimidine bonded together. Now the reason why adenine always bonds with thymine is because of the number of hydrogen bonds that can form between them. They can each only form 2, as opposed to guanine and cytosine, which can form 3. And then this is just kind of showing you, again, how it looks like a ladder. You might have to put your head on your side to see the ladder shape. But on the outside here and here, notice we have alternating sugar, phosphate. So sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, and so on. And then we have our base pairs in the middle. Now the double helix is said to be directional, and specifically, both strands are anti-parallel. Now here's that picture again. Now if you look at one side, it's upside down from the other. So here we have sugar, phosphate, but if we look at over here, and I'm actually gonna go backwards a little bit, you can see it probably a little more clearly in the previous picture. Notice how it's right side up here, and you can easily read it, but on the other side, it's upside down. Because imagine that these are the nucleotides. So we have a sugar, a phosphate, sugar, phosphate, and my fingers are the bases. If they were facing the same direction, I wouldn't be able to have the bases bond with each other. So one of them has to be flipped around, has to be flipped backwards in order for the bases to connect. So the strands run opposite each other because one of them is actually flipped around. It's upside down from the other. And that's just what we mean by directional and anti-parallel. They're both parallel in terms of, never touching, but they're anti-parallel because they are opposite each other. They're facing opposite directions. DNA is highly coiled and wound tightly around histones, forming something called nucleosomes, which then wind up into the chromatin. DNA is a really, really, really big structure. It is a huge macromolecule. And to keep it all nicely safe in the nucleus, we have to wind it up. And it takes a little bit of help to wind it up. So these individual little circles are called histones. They're a type of protein, and the DNA will wrap around the protein. The DNA wraps around the histones, and then when a bunch of DNA histone complexes wrap themselves up together, that's the nucleosome. So think of it like, I wish I had a piece of string right now. So my fingers are each histones. I wrap the DNA around it, wrap it around that histone, wrap it around that histone, wrap it around that histone, wrap it around that histone. Then I take all those histones, put them together, and coil that up. That is the nucleosome. And then we take a whole bunch of those nucleosomes and we supercoil them together, and that's how we get the chromatin. All right, let's talk a little bit about DNA replication. So it was Matthew Meselson and Franklin Stahl in 1957 showed that DNA replication was not conservative or dispersive, but rather it was semi-conservative. And we'll talk a little bit about what those two things, or what those three terms mean in a moment. So in general, what they discovered that the original DNA, the parent strands, separate. Each one is used as a template for the new strand, and then they'll stay with the new strand. So the resulting daughter DNA, the new DNA, always consists of one parent strand and one new strand. This is just kind of showing a setup of their experiment. You don't need to know that much about it from my class. But here, the red is the original parent. It separates, it goes through replication, you end up with two sets of DNA. Each one has a parent strand and a new strand. You can see how it gets passed on along. Let's talk about those three models. For a while, there was confusion as to how exactly they did this. So these were the three hypothetical models of how DNA was able to replicate itself. The first one said that in the conservative model, the parents separated, they were used as a template, and then the two parent strands came together and the two daughter strands came together. So here, they separate, they're used to create the daughter strands, then they come back together, and there's the daughter strands, and so on. Semi-conservative, the parent strands separate, they're used as a template, and they stay with the daughter strand that they help make. and so on and so on. Dispersive, to me, seems kind of crazy, but I'm just probably thinking too much about it. It's where essentially the DNA, the entire DNA gets chopped up. It gets, all the little pieces are used to replicate themselves, and then they get put back together in these random ways. Now, I'm glad that one model didn't stay too long, because in my mind, and maybe it's just because I've grown up knowing how DNA replicated, but it just seemed like that would take a lot of effort to make sure the DNA goes back together. And when you're talking about DNA being, 3 billion bases long, then that's a lot to keep track of. All right. Well, let's talk a little bit about the steps of replication. Well, first, let's talk about what it is. Replication is when we make another copy of our DNA. And we typically do this because we are going to get ready to divide that cell via mitosis. Anytime you go from one cell to two, you need to put the DNA in each one because it needs to know how to do its job. Now, the interesting thing about DNA is that it doesn't just start at one end and replicate all the way through. With how long our DNA is, that would take forever. So it actually replicates simultaneously at many different points on each chromosome. So many points on each chromosome, and don't forget we have 23 pairs of chromosomes. So we're talking about this is going on at hundreds of places all at once. So what that means is we'll form these little replication bubbles, and the replication fork is where we are separating the DNA. They'll expand, they'll work in both directions. That's what the arrows are telling you. That's replicating in that direction here, that direction. And all four of these, they get larger and larger. They open and expand more until they meet up. And then, look, wow, we have new DNA. Now, the first thing we have to do then is open up the DNA. Makes sense, right? We got to unwind it. We can't replicate it if it's all twisted up. And that's where DNA helicase comes in. It's the enzyme that's going to actually untwist and unzip the double helix. Then, once we have it opened up, we have an enzyme called primase. And at the initiation site, it's going to build a short RNA primer. Now, the reason why it needs to build this short RNA primer is because the next enzyme, DNA polymerase, needs to attach to something in order to build from it. So the RNA primer is simply a spot where the polymerase can now attach to and build the new strand. So let's see, here we have, I'm gonna move my camera for a moment, we'll look at this picture on the right. So this is the origin, helicase will unwind it like that, and then primase will come in, build these primers. Then the next part is DNA polymerase. It'll attach to the primers and start building them. Now DNA can only build in the 5'3" direction. Now what that refers to is 5' versus 3'. Those are looking at specific carbons in the sugar, deoxyribose, and which way they're facing. So DNA can only build the new strand in the 5'3" direction. So if you notice here, when it attaches there, it's building in the 5'3", so it can just keep following behind the helix, or the helicase, as opposed to down here, where it has to keep kind of jumping and playing essentially leapfrog. I do diagram that up on a board in one of my videos, which will be listed as a resource at the end of this chapter. I definitely recommend you check that out. Bases in the template and complementary bases in the new strand are hydrogen bonded together to form the new DNA helix. So as the DNA It attaches to the primer, and then it starts reading the original strand of DNA, the template strand. And it's going to build and attach complementary bases. So if it sees an adenine in the parent strand, it's going to attach a nucleotide with thymine. If it sees guanine, it will attach cytosine, and so on and so on. It's just going to keep reading the original strand, the template strand, and attaching the complementary bases as it goes. And it attaches the bases via hydrogen bonds with their complementary buddy. Here, this picture, we have helicase unwinding the DNA. Primase is building the primer. And I don't think I quite show polymerase yet. That's going to be next. So creating the new DNA. As we already said, priming, let's see, here we go. Helicase is unwinding it. We have primase building the primers. And then we have polymerases going along, reading the parent strand and attaching the new DNA strands to it, the new nucleotides. Now, as it does this and as it finishes up different sections, it's also going to go back and check for errors. And that's actually what DNA polymerase does. Its job isn't just to build the new strand and build the new nucleotides. Once it's done with that, it actually goes back and it proofreads its work. It checks to see if it made any errors. If it does find any errors, it will replace them with what should go there. It's also going to cut out those RNA primers and replace them with DNA bases, so actual DNA segments. Then we have our last enzyme that you need to know, ligase. Ligase is going to connect all the different parts where the backbone needs to fuse. So the sugar phosphate backbone, any place where they're not connected, ligase is going to make sure that they do join together. Replication is discontinuous, and this is kind of what we were saying about before. Because polymerase can only build in a 5-to-3 direction, that's going to cause one side to be slower than the other. Now remember, we opened up here, and then we kind of build as it continues spreading open. So as helicase is going here, we built the primer here thanks to primase. Polymerase attaches at this and is supply prime end, so it's going to keep building and following along in its 3' end. Now I know that this is 5' and this is 3', so if I look at the template strand, remember they're anti-parallel, so they should be different from each other. This should be a 5', this will be a 3'. 3', 5'. Not quite sure, these right here are incorrect. Now on this side, however, even though it's opening up that way, polymerase cannot build in that direction because again, it can only build in 5-3. So it'll attach here, build backwards. Once it reaches a place where it can no longer build, it disconnects, move to the next primer, build backwards. So it kind of leapfrogs over itself. And because of that, those Okazaki fragments, it does take that lagging strand longer because the polymerase has to keep disconnecting and reconnecting. Now we're going to finish up with just this overall picture. This is looking at, here was the origin of replication. We're only looking at this half. So we have 3' here, 5' on that end for the parent strand, which means that we are going to be building, if this 3' of parent end is the 5' of the new end. So the primer would have been built, helicase is busy opening up the DNA to the left, primase builds a primer here, polymerase attaches to it, and then it just continuously builds following the direction of the helicase. Because it can just go continuously, that's what we call the leading strand. However, down here, in the parent strand, we have 3' here, 5' here. Since 5' is here, that means that in the new strand, this is going to be 3', and over here is going to be 5'. And we can only build 5 to 3. So what happens is, you're going to have a lot more primers being built. So a primer was here, the polymerase attached to it, built backwards, 5 to 3, disconnected, jumped over to the next primer, built backwards, 5 to 3, disconnected, jumped to the next primer, built backwards, disconnected to the next primer. Oh, look, here's polymerase, builds backwards, and it's going to keep doing that. So once this polymerase is done here, it's going to disconnect and go to this primer. And it's going to keep doing that, leapfrogging along like that as it follows the helicase. Now, because we have all these fragments, we call them Okazaki fragments, and this strand takes longer to build because of the constant connecting and disconnecting of the polymerase. So we call it the lagging strand. Now, once polymerase is done, it's going to go back and he's going to take out these primers, fill them in with DNA nucleotides. And then ligase is going to connect all of those sugar phosphate backbones together to make a continuous strand. All right, so that's it for sections 2 and 3. I hope it was educational and informative for you. If you have any questions, let me know. Hit me up in Google Classroom or email. And in the meantime, you guys, stay awesome, stay curious, stay safe and healthy. All right, you guys, take care. Until next time.